Hello everyone, how are you all? I welcome you to the class of biology. Dear 11th class students, please report your attendance. Yeah. Centers, please report your attendance. Yeah, okay. Okay, so in the last class, I had given you an introduction about the various taxonomical aids and the living world reason for classification, the categories. Okay, so all of this I had done in the last class. So do you want me to give you a quick recap? Okay, I'll just check the chat. I believe it's not working a bit. Okay, so all right, in the meantime, let me give you a quick recap of the chapter. The living world, what we have covered so far. In this chapter, we had studied that there are 4 basic properties of living organisms, and these properties are basically growth, reproduction, metabolism, and consciousness. Okay, these are the 4 basic properties which bind all the living organisms together. Okay, so growth, reproduction, metabolism, and consciousness. Everybody understands this? Yeah, okay. Shillong, thanks for your attendance. Other centers, please also report your attendance. Okay. Yeah, as soon as you join, just report your attendance. All right. So when we talk about growth, I want to talk about the various processes that happen in the body, but it's a very simple definition that increase in mass is called as growth. And after that, there is the property called reproduction. The reproduction property, it is the ability of an organism to make organisms similar to itself. New organisms which are similar to itself, it is called reproduction. And growth and reproduction, they are not defining properties, they are basically the properties which are shown by living organisms, but not all living organisms, or due to the fact that non-living objects also show growth, these properties are not considered as defining properties. Metabolism and consciousness are both considered as living organisms' basic properties or defining properties. Metabolism is the sum total of all the metabolic reactions, the chemical reactions that happen within the cell. Consciousness happens to be the ability by which an organism reacts to its stimulus. All right, so these are the basic 4 properties that we have been talking about. Regarding growth, reproduction, metabolism, consciousness, if I am saying that metabolism and consciousness are defining properties, this means that if I encounter an unknown biological material, like for example, you come across venom, okay, so venom-like material in outer space, okay, somewhere, and it shows consciousness. Venom shows consciousness. Everybody must have seen the movie Venom, right? If you have not seen the movie, you must have seen the trailer. It's a science fiction movie and it's a very nice one. Or if somebody goes on planet Mars and finds an unknown biological material which is reacting to touch, okay, trying to kill us or trying to be friendly with us, anything can happen. So that can be said to be a living material, okay, because it has consciousness. And if you want to see it closely, it might show some heat pattern, heat release or heat absorption capability. So it should have another property called consciousness. Based on these 4 properties details, I can say that viruses are not living organisms. They are between living and non-living, okay, because they show growth, reproduction, but no metabolism of their own and no consciousness of their own also. So they are not considered as living also, but we cannot consider them as non-living also. So that is the basic thing about these 4 properties. Another important thing regarding consciousness, consciousness is found only is found on in only living organisms, but most advanced form of consciousness called self-consciousness. This property is a characteristic feature of human beings only. Not all organisms have self-consciousness. Only human beings have self-consciousness. Then after moving to the other things in this chapter, I came across taxonomy, importance of taxonomy. Why do we classify organisms? I explained this concept to you, that we classify organisms because it becomes easier to study them. We give classes or names or nicknames to other people, and when we talk about this practice of giving nicknames or classifying people around us, it is also seen in the scientific classification of organisms, and we do this so that it can be easier for us to study them as large categories. If we see an unknown spider-like organism with 8 legs, we will call it a spider. Okay. If I see a snake-like thing at night, I will call it a snake, right? So there are some basic things which identify or distinguish these organisms from other organisms. Okay. So classifying is having this purpose. So classification, taxonomy, identification, all these things will happen. Then I gave you the rules of taxonomy given by Carolus Linnaeus, called the binomial nomenclature system and the rules of nomenclature. After that, we had discussed our taxonomical aids: botanical garden, zoo, herbarium, museum, and key. All these things we had done in the last class. Now in the present class, I had asked you, in the last class I had finished this chapter. Now in chapter of biological classification, I had asked you to do some homework. I had asked you to write down the Whittaker's, R.H. Whittaker's criteria for classifying organisms into 5 separate kingdoms. Have you done your homework, students? Have you done your homework? I asked you to write some parameters for classifying organisms according to Whittaker. So what were the parameters used by Whittaker? Do you remember that? Yes, anyone? Do you remember those parameters? Nobody? Okay, nobody remembers those parameters. You can try. Alright, no answer. Okay, I'll do this for you. Look, biological classification is what we have to deal with now. Biological classification is what we are dealing with now. And in biological classification, the chapter is, please put the heading, the biological classification. Biological classification. Okay. So I had told you that the 5 kingdom system was given by Robert Whittaker. Aristotle gave the first scientific classification system, followed by Haeckel, Copeland, and finally Whittaker. Aristotle, I told you about this, Aristotle's system of classification. Herbs, shrubs, trees, and plants were classified like this. Animals, Anaima, Enaima, gave the first scientific classification system. First scientific classification system. But after Aristotle, there were other classification systems as well. But the most accepted one is the Linnaeus system of classification. Linnaeus gave 2 kingdom system of classification. 2 kingdom system of classification. Hackle gave 3 kingdom system. Copeland gave 4 kingdom system. And after this, the one we have to study is Whittaker's system. Whittaker's system of classification, and this is the 5-kingdom system of classification. This is the 5-kingdom system of classification. Now, what are the criteria used by Whittaker? Criteria used by R.H. Whittaker for classifying organisms were: number 1, characters used were cell type, whether eukaryotic or prokaryotic. Second criteria was whether cell wall is present or absent. Third is nuclear membrane present or absent. Nuclear membrane present or absent. Then comes body organization. Body organization. And fifth character is mode of nutrition. Mode of nutrition. So these are the 5 criteria. I told you that there is a table in your NCERT, you have to learn it. Okay. So how do we classify the organisms into 5 separate kingdoms? This is according to that part. Okay. All right. So dear students, now we will have some other details regarding Whittaker's system. Whittaker gave the 5-kingdom system of classification, but even so, nobody was fully satisfied. There were some other scientists like Carl Woese, and Woese said that 6 kingdoms should be there, not 5, because archaebacteria are a new type of, it's a separate category, archaebacteria, okay, and these are the most ancient organisms. So, because you have not drawn this table by yourself, I have to do it for you. You will draw a table like this. Here you will write characters. Here you will write characters. You will write here cell type. Okay, there's a honeybee in here. Okay. All right. Oh no, sorry. It's a large size housefly. Mega size, okay. Anyway, so the 5 kingdoms are here like this. Okay, so 5 kingdoms are cell type like this. So based on the cell type, we have some other things here. Wait a second. Okay. So first character is cell type. Kingdom Monera, the cell type is prokaryotic. Prokaryotic. Let me use a different color for this. Prokaryotic. This table is very important for your boards because it's frequently asked. All the other kingdoms except kingdom Monera, they are eukaryotic. Monera, Protista, fungi, plantae, and animalia. Monera, Protista, fungi, plantae, and animalia. Okay. So Monera is prokaryotic, all the other kingdoms are eukaryotic. Eukaryotic. Okay, this is also eukaryotic. So all the organisms have now been classified based on the cell type. So now we got our first kingdom based on cell type. We have differentiated Monera from the other kingdoms, right? Okay. Then the other character is cell wall. Cell wall. Okay, and cell wall, and the cell wall is non-cellulosic. Non-cellulosic, polysaccharide plus amino acid. Actually, there are two things here. N-acetyl glucosamine and N-acetyl muramic acid are the basic things that are required, right? Right, so yeah. Okay, so these are the things that are used to make it. This is the cell wall of bacteria. Now one more thing. First of all, try to understand this. Cell type, prokaryotic, eukaryotic. Because the prokaryotic cell is very different from eukaryotic cell. You know this because prokaryotic cell, the bacteria, they have no nucleus, they have no nuclear membrane, they have no cell organelle except ribosome or maybe flagella. So they have very less structures, few structures. Also, prokaryotic cell is very small compared to eukaryotic cell. Prokaryotic cell is very small. So based on these things, I have separated a kingdom called Monera. If it is prokaryotic, it has to be a bacteria. It has to be a moneron. Now, Monera not only includes bacteria, it includes other organisms also. If it is eukaryotic, it can be Protista, fungi, plant, animalia. Now, the second character is cell wall. Based on cell wall, you can say that bacteria has cell wall. Protista might have cell wall, Protista might not have cell wall, so we cannot say definitely. Fungi has cell wall, plants have cell wall, but animal kingdom does not have cell wall. Okay, so based on this character, we have separated animal kingdom also. In animal kingdom, cell wall is absent. In all the other kingdoms, what is the status? Cell wall is present in some, absent in some. So I will just say present in some. It is present in fungi. Okay, and in fungi, plant, the cell wall is made up of chitin, N-acetyl glucosamine polymer. In Plantae, it is also present, and here it is made up of cellulose. Okay, here it is made up of cellulose. Here it is made up of cellulose. Okay, yeah, I'm a bit uneasy. Yeah, okay, all right. So hope this does not happen to you often. All right. After that, we have a new character called the nuclear membrane. Nuclear membrane. Okay. The nuclear membrane is absent in Monera. It is prokaryotic. Protista, it is present. In all kingdoms, it will be present, except Monera. You can also put a tick sign here to see if it is present or absent. Okay. Then the next character is body organization. Body organization. Like this. So based on body organization, what I can say is, in prokaryotic, it is only a single cell which is forming the body. So you can say it is cellular body organization. In eukaryotic, in Protista, cellular body organization. But in case of fungi, you will say it is multicellular, not a single cell, but a multicellular structure. Multicellular structure or loose tissue. In Animalia and Plantae, it is tissue organ. Plantae is tissue or organ based. But in animals, these are very specialized, so I call it tissue organ or organ system. Tissue organ or organ system. The last criteria, according to Whittaker, it is called mode of nutrition. mode of nutrition. Mode of nutrition. Okay. So in case of mode of nutrition, in bacteria it is autotrophic. Right? In bacteria it is autotrophic. In eukaryotic protista it is also autotrophic. Now I need to distinguish it here. Protista has most diverse form of nutrition. So I can also call it autotrophic plus heterotrophic. Same is the case with bacteria. They are autotrophic, okay, and heterotrophic. Same case here. Okay. In case of fungi, it is heterotrophic exclusively, because fungi don't have chlorophyll. In plants, it is exclusively autotrophic. In animals, it is heterotrophic with holozoic or saprophytic nutrition, etc. Heterotrophic, that is holozoic or saprophytic, etc. Okay, this is the thing. All right. So he proposed a 5-kingdom system of classification. Okay. So please try to remember these criteria. This one, this one, this one, this one, this one. So these are the criteria used by Robert Whittaker. Right. So if anybody asks you, you will be able to tell the criteria used by Robert Whittaker. But this is not the end of the story. After Robert Hooker gave the system, there were many people who said that I can give a better system of classification, right? So everybody was not convinced. Among one of these scientists was Carl Woese. Carl Woese studied these results for many, many years, and he came to the conclusion that this is not a sufficient system. The 5-kingdom system is not sufficient. There should be a new kingdom called the 6th kingdom, and archaebacteria should be placed inside. Okay. So thereafter came the 6th kingdom classification system and the domain system. Sixth kingdom system. It was proposed by Carl Woese. It was proposed by Carl Woese. According to him, archaebacteria are different from other bacteria So these must be placed in a separate kingdom. So these must be placed in a separate kingdom. This was what Carl Woese was doing. He also gave the concept of 3 domains of life. Please finish writing. Please finish writing quickly. Then we'll start with kingdom Monera. Should be placed in a separate kingdom. Right? All right. He also gave the concept of 3 domains of life. Carl Woese gave the concept of 3 domains of life. Gave concept of 3 domains of life. What are these 3 domains? These 3 domains are archaea, prokarya and eukarya. Archaea, prokarya, and eukarya. Now in archaea, all bacteria will be ancient bacteria will be included. In prokarya, all the normal type bacteria will be included, and eukarya will include all the organisms that are including humans. Okay. Yeah. So let's move on quickly. I hope you have finished it. Now my question to you is, your homework, Carl Woese compared dash S unit of ribosome okay. So you have to tell me which unit of ribosome was compared between different organisms. Different organisms. So you have to tell me what type of ribosomal RNA or messenger RNA, whatever that was, that helped the ribosome to identify its target. Okay, this is the reason. Now you can properly start with Whittaker's system of classification, and the first topic in this regard, it is called kingdom Monera. We are going to study Whittaker's system only. For some question purposes, you can use that so and so. All right. So now please tell me, what are bacteria being classified on the basis of? Can you tell me, how do you classify bacteria? Any idea to anyone? Raise your hands. If you cannot, come to the keyboard. No hands on the shape. Yeah, shape is one criteria. Shape is one criteria. We classify them on the basis of multiple criteria: shape, size, flagella or no flagella, how many flagella, okay, things like this. So please write this. The kingdom Monera includes bacteria, okay, and archaebacteria. Archaebacteria. So first of all, we will complete and see what archaebacteria are all about. So please put the heading: archaebacteria, etc. What are archaebacteria? Right. So now please tell me, what is the unique thing about archaebacteria that differentiates them from other organisms? Look, there are so many things that are not mentioned in NCERT and very important for your boards. Archaebacteria, they have a lot of similarities with they have a lot of similarities with bacteria, some similarities with eukaryotes, and this indirectly means that archaebacteria have given rise to or are the ancestor of both human beings and all organisms, and including bacteria, because archaebacteria found much before bacteria were first born. So kingdom Monera includes bacteria and archaebacteria, etc. Archaebacteria are the ancient bacteria ancient bacteria. Ancient bacteria will be sufficient. It's not necessary to write karyotic or not karyotic. Ancient bacteria, because they have not changed one little bit. So archaebacteria have been classified archaebacteria can be classified on basis of habitats. On the basis of habitats. So we have thermoacidophile, we have methanogens, so many different types of archaebacteria. Classified on the basis of habitats. So what are these? First category is methanogens. Methanogens, as the name suggests, they are methane producing bacteria from organic matter. They produce methane gas from methane loving bacteria. sorry, just a second. They produce methane gas from organic matter. Organic matter. Now I will tell you an interesting story. In Delhi, there is an engineering college, and just in front of that engineering college, there's a big sewage water system. By big sewage water system, I mean it's a canal with lots and lots of sewage water. So one person who was studying in the engineering college got an idea that if I'm experiencing so much of bad smell from this dirty water source, why not make some good use of it? So what did he do? He developed a technique he developed a very simple instrument, just using iron pieces welded together and drums welded together, and he used it to make a gas collection chamber. Okay, and in this gas collection chamber, it is so strong that for the past 3 years, he donated this to a tea seller. The student donated this instrument to a tea seller, and he says that for about 3 years, I'm not getting any, I can burn this continuously, and even after 3 years, there is no problem. So methane gas is produced from organic matter. Now, how many of you have been to old archaeological sites, old tunnels, old wells or something like that? Have you seen some warning in newspapers that a person went into a cave and he could not come out, he was paralyzed, or his life was saved with great difficulty, or he died due to suffocation or some other reason? So why do these things happen? The reason is methane gas. Methane gas producing bacteria can live only in anaerobic conditions. Only in pure anaerobic conditions they can live. They are found. What is their habitat? They are found in various places okay. Example, rumen of cow, stomach of herbivores, okay. So they are found in various places like this. All right. The next thing is they are also used. Look, habitat is done. They are obligate anaerobes. That is, they cannot grow in the presence of oxygen. They require anaerobic environment. So if we cannot give them anaerobic environment, they will ultimately die. Obligately anaerobic or anaerobes. Obligate anaerobic or anaerobes. One more thing. Now I must come back to the question. Regarding very old tunnels, very old mines, very old passageways, old temples, and in some forts there are some secret passageways that the government does not want you to enter. Have you seen these kind of things? Why does the government not want you to enter these places? Anybody? Just raise your hands if you can guess the answer. You must have been to many places where there is a government warning that please do not enter this area due to so and so reason. So what is the reason I am asking you? Because these places release a large amount of methane. So if anybody goes near it, he might be paralyzed. Methane has this property of inducing anesthesia in people. Okay. So a lot of people on inhaling methane in large amounts, they can become unconscious and they can be prone to attacks or something. So many things can happen. Right? He can be brain dead for life also in extreme case. So methanogen is the first category. Example of methanogens are methanobacterium, methanococcus, etc. Methanobacterium, methanococcus, etc. All right, this is all about methanogens. Second category is halophiles, salt-loving bacteria. Okay, what is halophile? It's a salt-loving area, a salt-loving organism. These are found in extreme salt concentration from 10 to 30% NaCl. Okay, extremely salty concentration. These areas, the halophiles are present. It contains a large amount of sodium chloride also. Right. So loop of Henle, I am saying. Halophiles, they are found in extreme salt concentration. If salt concentration becomes less than 10%, they will stop growing. They will immediately stop growing if it encounters an area less than 10 centimeter of rainfall. So these are halophiles for you. Okay. The third category, examples first, right? Halobacterium, halococcus, etc. It's easy to remember these names. So halobacterium and halococcus. Etc. There is one more interesting bacteria in this category. Pseudomonas putida is a bacteria which is having the habit of clearing up oil spills. Oil hydrocarbons, it can easily convert them into carbon dioxide. So this bacteria becomes very handy in those situations. All right. Halobacterium, HALO, HACTERIUM. Okay. This is Halobacterium, halococcus. The third the third category of archibacteria, I hope everybody has written it. Okay, that is the methanogen is done, halophile is done, thermoacidophiles. Thermoacidophiles. Thermoacidophiles, meaning they love hot, salt, and acidic conditions. Okay. Halophile done, methanogen done, right? So this is the last category. Halobacterium is done, halophiles is done, thermoacidophiles. Thermoacidophiles. These are those bacteria which can survive only in high concentration of acid or something like that. Thermoacidophiles, please write. Um, these bacteria are found in very extreme regions of the earth. These bacteria are found in very hot acidic conditions on earth. Okay. Example, thermococcus, thermobacterium, etc. I hope you understand this, right? In this case, methanogens found in stomach of herbivores, halophiles found in salt concentration control and other things. Thermopseudophores are found in very hot basic acidic conditions on earth. Okay. Yeah. Example, thermococcus, thermobacterium, etc. Okay, yeah. Students, do you need a break now? Because it's nearly time for break. I think you should take a quick break now, and after that we'll, yeah. So yeah, I have to do some gram staining thing also. Yeah. So after break I'll discuss this. All right. So yeah, everybody needs a break, right? Yeah, because we are 1 hour. We have successfully spent 1 hour, right? So take a quick break, and after break I'll discuss this, why these bacteria are so much responsible, so many things. I have to explain a few ecological disasters also. Okay. All right, so we are back. Dear students, okay. So Shillong students, we were talking about archaebacteria. So please take a quick look. Archaebacteria classified on the basis of their habitats, that where are they found. So methanogens bacteria, these are found in some places which are having anaerobic environment, and wherever you can see the smell of methane is associated with rotting organic matter. So whenever you see such places, it is quite probable that these are due to methanogens bacteria. In gobar gas plant, in the sewage gas treatment plant, the gas that is coming out, it is methane. So I told you that there are some notices at very old monuments. There are some secret passages, secret canals that have been closed by the government. The reason is that in these places, methane gas is found. If we inhale methane for a large amount of time, we might become unconscious. Okay, everybody falls unconscious, and that is the reason why people die in such places. In the cases of, you must have read a lot of news that a sewage worker, a person who cleans the sewage, was working without the proper safety equipment, and he fell into the sewage drain or something like that. These things happen in big cities, and in small cities also. It is not uncommon, not very common, but it's not uncommon also. So sometimes such unfortunate incidents happen, or someone was drawing water from an old well. In all these places, in these old structures, there are some organic matters, and there is anaerobic environment. In these conditions, methane is formed, and this methane might render a person unconscious, just like chloroform. So they might become easily, they can fall easily, and over long period of time, methane is poisonous. If somebody keeps on inhaling methane for a long amount of time, even though he is paralyzed, he is still alive, but if he does that for a long amount of time, he will die. All right. This is our methane. Now halophiles are found in extreme salt concentration. Okay, if salt concentration falls below 10%, these bacteria will die. If salt concentration falls below 10%, they will die. So they can survive in extreme salt concentration only. Okay, if it is below that, they will not survive. Example, halobacterium, halococcus. Then there are thermoacidophiles. Thermoacidophiles are found in very hot acidic conditions on earth. Boiling acid lakes, boiling lakes of 80 degrees centigrade to 100 degrees centigrade, lake containing hot sulfuric acid. In these conditions, nobody can survive, but there are still some bacteria which can survive under this extreme condition, and these are called thermoacidophiles. So it's truly unbelievable, but it's true. Example, thermococcus, thermobacterium, so many other bacteria, right? So now the question becomes, we have classified the archaebacteria. So the basis of classification was habitat, and these are found in very extreme environments. Now you will make a point. Archaebacteria are able to survive in extreme environments due to their cell wall. The cell wall is unique and very resistant. That is why they are able to survive in such extreme environments. Also, archaebacteria have a unique cell wall. Which is made of monolayer of lipids. Monolayer of lipids. In all other organisms, bilayer is found, but in archaebacteria, monolayer is found, right? Monolayer is found, all right? So they have a unique cell wall which is made up of a monolayer of lipids. Archaebacteria have a unique character which is not found in other organisms, So what is this character? Archaebacteria have a monolayer of lipids. This is a unique character because this is not seen in any other kingdom. Unique. Now there is one more thing. Archaebacteria they have a unique cell wall. This is made up of a monolayer of lipids. And one more thing, archaebacteria have histone proteins. Histone proteins. Do you people know about histone protein? Histone protein, does anybody know? Yes or no? Raise your hands if you know about histone protein. Nobody? Okay. Histone protein is a protein which is used for packing of DNA in eukaryotes. So this character is similar to eukaryotes. This proves that archaebacteria are ancestors of eukaryotes. If it was not true, then this would not have been the case. Histone proteins. Histone proteins are found in eukaryotes only. Histone proteins are found in eukaryotes only. Found in eukaryotes only for packing genetic material. The purpose of it is packing genetic material. So they have histone proteins and these are for packing the genetic material. But this is a character of archaebacteria also and eukaryotes also. So this means that eukaryotes have evolved or they are the ancestors of, the archaebacteria are the ancestors of eukaryotes. Okay. Archaebacteria do not have nuclear membrane, and have unpacked genetic material, unpacked nucleus or primitive nucleus. Just write primitive nucleus, it is sufficient. Do not and have primitive nucleus. Okay. So this is the thing. So this is the similarity with prokaryotes. This is their similarity with prokaryotes. Okay. This is their similarity with prokaryotes. Okay. All right. Next thing is their similarity with eukaryotes is this thing, histone proteins. So archaebacteria have similarity with eukaryote, prokaryote, and they have unique characters. Similarity with eukaryotes. EK I am writing. EK means eukaryotes. Okay. So these are the important things about archaebacteria. We call them archaebacteria because archaebacteria have not evolved from very, very long period of time. Millions of years ago, they were the same as they are today. Okay. That is why we believe these are the most ancient organisms on Earth. Okay. Their fossils have been found in very, very old rocks. All right. So this is about archaebacteria. Now please put the heading eubacteria or proper bacteria, true bacteria. Eubacteria or true bacteria. Eubacteria or true bacteria. So what are these bacteria doing? What are these bacteria? Where are these bacteria found? Habitat and habit. Anyone? These are found everywhere. Eubacteria are cosmopolitan. This means they are found everywhere. Okay. They are found everywhere. Okay, so soil, water, land, earth, air. Okay, even in the upper layers of atmosphere also some bacteria are found. So they are cosmopolitan. Okay, and we have classified these bacteria. Okay, and because of their habitat, their habit, a lot of things they have been classified also. Please write now. This is their habitat. Habitat is everywhere. Habit, eubacteria are mostly heterotrophic. Mostly heterotrophic. Okay, this means that they mainly have a habit of eating other organisms by being parasitic or by feeding on dead organisms or dead organic matter, but sometimes they may be autotrophic also. So this is their habit. This is their habit. Okay, they are mostly heterotrophic. Next question is, if they are a heterotrophic, this is done. Habitat is done. Habitat is done. Now we have to classify. Classification of bacteria classification of bacteria on basis of size and shape. Size and shape. On basis of size and shape. So on the basis of size and shape, if it is spherical shape, it is called coccus. Okay. If bacteria is spherical, it is called coccus. Now, if it is singular, it is called monococcus. Okay. If it is double, it is called diplococcus. If it is forming a chain of bacteria, this is called streptococcus. And if it is forming a bunch of grape-like structure, it is a very dangerous bacteria of this category. It is called staphylococcus. It is very hard to treat by antibiotics. It is called staphylococcus. Okay. Coccus is this. Next there is the rod-shaped bacterium. It is called the bacillus. If it is singular, it is called monobacillus, rod-shaped. If it is singular, it is called monobacillus. If it is forming a chain-like structure like this, it is called streptobacillus. Okay. Now, if it is having a rod-shaped structure like this, it's called vibrio. Sorry, it's comma-shaped. It is called vibrio. It's called vibrio. Right. Now, if it's having a spiral shape like a corkscrew, it's called spirillum. Corkscrew or spiral shape. It is called spirillum. All right. Okay. So these are the various shapes of bacteria. These are the various shapes of bacteria. Now there is one more criteria based upon classification of bacteria based on flagellum. Based on flagellum, if a bacteria has a single flagellum, it is called monotrichous. If flagellum absent, it is called atrichous. Okay. Now, please, have you written this? Have you written this? Yeah, please do this quickly. Yeah. All right, done. Okay. So if this is like this, it is called monotrichous. Now, if bacteria has flagellum on both ends like this, it is called amphitrichous. Amphitrichous. If bacteria has flagella on both sides in bunches like this, then you will call it, sorry, just a second. Just a second. This is amphitrichous, flagella at both ends. Yeah, if flagella has, if there is a group of flagella at one end, this is called lophotrichous. Lophotrichous. Okay. If there are flagella all over the surface of the bacterium, it is called peritrichous. Peritrichous. Okay. Atrichous, monotrichous, amphitrichous, lophotrichous. Okay. So these are the various types of flagella characters. Okay. All right. Now there is one more criteria. Please finish it. I hope you have done it. Now there is one more criteria based upon gram staining. Gram staining. All right. Now, gram staining is a very important technique. Gram staining is a very important technique. Gram staining is a very, very important technique. All right, so let's do it. How do you understand gram staining? So let me tell you an example. For understanding gram staining, I will take a glass slide. Okay, I'll take a glass slide and I will spread bacteria over it. How do I spread bacteria? The technique for spreading is like this. I take one glass slide and I take another glass slide. Then I rub these together. I take this edge and I try to spread the bacteria over all the surface of the glass slide. Now I heat it over a candle flame or a gas burner flame. Heating means not destroying all the bacteria. I just want the water to evaporate very quickly, so that when the water evaporates in this water film containing bacteria, now the bacteria will stick to the surface of the glass. Bacteria will stick to the surface of glass. So I will just take it over quickly over the flame, and its water starts evaporating. I will quickly remove the glass slide. Okay. Now this glass slide is having fixed bacteria. They will not leave the surface of the glass slide until and unless I scrub it with a Scotch Brite scrubber or something like that. Okay. Now I put some, or there is a stain called crystal violet. So suppose this is orange colored. This looks like eosin, but you must imagine this is a violet colored, blue colored bottle. Now I will take some stain from it. I will pour it on this glass slide, and I will spread the stain all over the glass slide. And then after that, I will take alcohol and I will pour over the glass slide. Alcohol will cause excess of stain to be removed from the glass slide. Now if I take this glass slide under the microscope, I will see some bacteria have retained the color of the previous stain, that is violet colored, and some bacteria have become colorless. Now how do I stain the colorless bacteria? I will take pink colored stain called safranin, and I will put some safranin over it and then wash it with water. When I see this glass slide now on the surface on the microscope, I will see pink colored bacteria and violet colored bacteria. The violet colored bacteria are called gram positive because they have retained the gram stain. The pink colored bacteria are called gram negative because their stain was washed by alcohol. So they have to be stained with a different thing. This is called gram staining. So if we find that on the basis of gram staining, there are only two groups of bacteria, one which catch the stain, one which do not catch the stain. These are called gram positive and gram negative bacteria. So please write now. HC Gram developed a technique, staining technique, to differentiate between bacteria. So first I will take a glass slide. I can label it glass slide containing fixed bacteria. Containing fixed bacteria. Okay. Now on this glass slide, I will put some crystal violet stain. I will use crystal violet color also. This is the crystal violet color. Everybody can see this color, right? This is crystal violet color. When I use this crystal violet color, the bacteria, all the bacteria actually, they will become violet in color. Okay. Now I wash it with alcohol. Alcohol wash. It will remove the excess stain. And now I can see some bacteria have become colorless, while others have retained their stain. Some bacteria have become colorless. So colorless I am denoting by sky blue color, like this. Some bacteria have become colorless. So these are colorless bacteria. And these bacteria are violet colored bacteria. Okay. All right. Now I will do one more thing. I will add a second stain now. It is called saffronin. A pink colored stain I am going to add here. And the colorless bacteria, they will suddenly become colored. Okay. So these bacteria, which were colorless, now they have become pink colored. Okay. This bacteria, it is called gram-negative bacteria. Gram-negative bacteria. This other bacteria, it is called gram-positive bacteria. Gram-positive bacteria. All right. So on the basis of gram staining, I am going to differentiate that there are two types of bacteria. Either it is gram-positive or gram-negative. So please finish this quickly. Let me know when you have finished. Done? Okay. Now let's write a few notes about gram staining. Gram used a stain called crystal violet, now called Gram's stain. It is now called Gram's stain, called crystal violet. To stain the bacteria on glass slide. on glass slide. All the bacteria become violet colored or purple colored. All the bacteria become violet colored. Now, alcohol is added to remove extra stain and some bacteria become colorless by alcohol. Some bacteria become colorless by addition of alcohol. Alcohol means ethyl alcohol. You can also use isopropyl alcohol or methanol. Normally ethyl alcohol is used. All right. Next point. Now, a second stain called counter stain is used to color the colorless bacteria This stain is saffronin. This stain is saffronin, and bacteria become pink in color. Bacteria which were colorless become pink in color. Okay. So these bacteria are called the bacteria which retain the stain are called gram-positive, which retain the stain are called gram-positive. The bacteria which are pink colored because they have become colorless, then they become colored by a different stain, which are pink colored, are called gram-negative. Now my question to you is, why would this happen? What is the difference between gram-positive and gram-negative bacteria? This is my question. The answer will be gram-positive bacteria, they do not have lipids in their cell wall, or very few lipids in their cell wall. But gram-negative bacteria, they have a lot of lipids in their cell wall, and their cell wall is very thin. So their stain is washed away by the alcohol. The lipids and the stain both are washed by the alcohol, so that is why they become colorless. Okay. The reason why gram-negative bacteria become colorless is because they have a high percentage of lipids in their cell wall, in their thin cell wall. So they lose the stain and their cell wall both go away with the addition of alcohol. So they become alcohol washes away the lipids along with stain, because stain is also washed along with lipids. So that is the reason why this is happening. All right. They have a high percentage of lipids in their thin cell wall, and alcohol washes away lipids along with the stain. Gram-positive bacteria do not have lipids in their cell wall. So your homework, it is going to be differences between gram-positive and gram-negative cell wall. Gram-positive and gram-negative bacteria. You have to write at least 5 differences. This is your homework. I will do up till this only today. And after this, we will meet in the next class, okay, which is the next week. So this is your last question for today. Differences between gram-positive and gram-negative cell wall. All right. So in the next class when we meet, I'll show you a few pictures regarding the archaebacteria. There are a lot of things to show. So in the next class, we'll deal with this. Okay. And in the previous class also I had covered a few topics. Your test, that will be in the following week. It will be consisting of living world. So prepare well. There is not much details in that chapter. It's very easy. Just remember what I have taught you and follow the module, and you will very easily be able to do that. So thank you, dear students, and thank you for your time. Good night, and travel home safely. Okay. So dear students, we'll meet in the next class now. Thank you.